all for joining me. Now, in the last lesson, we went over the eukaryotic cell, how it differs from the prokaryotic cell, and many of the features that were seen in said eukaryotic cells. This lesson will be more into what goes on inside and inside certain functions of the eukaryotic cell. As I said in that previous lesson, a eukaryotic cell has organelles inside it that prokaryotes do not. Does not say prokaryotic cells are simple, as they are not, but eukaryotic cells are extremely complex comparatively. And eukaryotic cells are kind of like their own creatures in a way, as they have their own quote-unquote organs, called organelles, that have certain functions, similar to how animals or humans have organs inside them that serve certain functions. So this lesson will be over the endomembrane system and the proteins inside it. We went actually over most of the organs in the previous lesson anyway, but this is more of a rundown of the endomembrane system, basically what's inside a membrane. So let's get into it. The endomembrane system, remember endo means within, is a group of membranes and organelles in eukaryotic cells that work together to modify, package, and transport lipids and proteins, lipids being fats and proteins being proteins, which are the building blocks for amino acids. It includes a nuclear envelope, lysosomes, and vesicles, which we went over in the last lesson. So if you want to go over that again, I suggest you watch the previous lesson. Although not technically within the cell, the plasma membrane is included in the endomembrane system because, as you will see, it interacts with the other endomembranous organelles. Now, the endomembrane system does not include either the mitochondria or the chloroplast membranes, which again we went over last lesson. Alright, here's essentially the system. You have the Golgi complex. You have, what is this again? Oh yeah, there's the rough epidermal, it's called, and the smooth. I can't remember what it's called. Like, I forgot what the R was. But you have the rough ER and the smooth ER. You have the cis face and you have the trans face, and all this will make sense once we go over it. So let's get into it. Reticulum. I always want to say rector or something like that, but it's not that. Anyway, first we have the endoplasmic rector, which is essentially this whole thing. Like the smooth one is here somewhere. I think, well, let's go over it. The endoplasmic reticulum, or the ER, is a series of interconnected membranous sacs and tubules that collectively modifies proteins and synthesizes lipids. However, these two functions take place in separate areas of the ER, the rough ER and the smooth ER respectively. What this essentially means is that it doesn't create proteins, it just changes the proteins that are created. But does create lipids, or can create lipids. We call the ER tubules hollow portions the lumen or cisterol space. The ER's membrane, which is a phospholipid bilayer embedded with proteins, if you remember a couple lessons ago when we went over lipids, phospholipids basically means waterproof, is continuous with the nuclear envelope. So first we have the rough ER. It is called a rough ER because the ribosomes attached to its cytoplasmic surface give it a studded appearance when viewing it through a microscope. Like here, on this, okay, I see. On this side, it's gonna be rough because there's all these. What did I say? Yeah, sorry, ribosomes. My mind's not working very well today. Has all these ribosomes on the outside. On the other side of it, it's kind of hard for me to like if you were to flip it, you would have the smooth that does not have ribosomes on it. So, but we'll talk about that in a minute. Ribosomes transfer their newly synthesized proteins into the rough ER's lumen, where they undergo structural modifications, such as folding or acquiring side chains. These modified proteins incorporate into cellular membranes, the ER or the ER's or other organelles membranes. The proteins can also secrete from the cell, such as protein hormones and enzymes. The rough ER also makes phospholipids for cellular membranes. If the phospholipids or modified proteins are not destined to stay in the rough ER. They will reach their destinations via transport vesicles that bud from the rough ER's membrane. Let's see here. Figure 1-4. Okay, here we go. Okay, so right around here. Since the rough ER is engaged in modifying proteins, such as enzymes for example, that secrete from the cell, you would be correct to assume that the rough ER is abundant in cells that secrete proteins. This is the case with liver cells, for example, and that's very true. Your liver has, the liver cells have an abundance of rough ER secreting proteins. That's part of the reason why it can repair itself, and it can I said nothing to repair, I guess, but it can, oh no, it does. It can also discharge toxins. That's why your liver can repair itself and remove, like, the toxins when you drink alcohol or whatever, or anything really. Then we have the smooth ER. The smooth ER is continuous with the rough ER, but has few or no ribosomes in its cytoplasmic surface. The smooth ER's function includes synthesis of carbohydrates, lipids, and steroid hormones, detoxification of medications and poisons, and storing calcium ions. In muscle cells, a specialized smooth ER, or sacroplasmic reticulum, is responsible for storing calcium ions that are needed to trigger the muscle cell's coordinated contractions. And that's also true. A lot of muscles, not all muscles I don't think, a lot of your mechanical muscles, or kinetic muscles as some people call it, the cells have abundance of smooth ER to help store energy. When we get into energy, there are basically 3 types of energies. Just a little bit of preview. You have like your instant energy, your what's it called? Your storage energy, and then your emergency energy. When you have energy, and when you eat and you store energy, your body will first bring energy from the muscles, not the muscles themselves, but from the muscles to get you moving. That's where the smooth ER comes from. If you don't have a whole lot of, if you run out of energy from that source, it will go to your storage energy or your fat. A lot of times, a lot of people, basically when you output more than you're inputting, you lose fat. That's essentially what exercise is. But it's also storage in case there's a famine or whatever, or you can't eat right away. So first you'll go through your instant energy, then you'll go through your storage energy. Then, if, heaven forbid, you run out of storage energy, then the muscles and stuff will break down themselves. Generally, you don't want to get to that point, obviously. But that's essentially, I guess that's kind of a preview when we get into metabolism and stuff, that the reason that can happen is because of the smooth ER, because it can hold things like calcium and stuff. Alright, here we go. Then we have the Golgi Apparatus. I've also heard it called the Golgi Complex. That's why I called it that earlier. In fact, when I was learning biology, they actually called it Golgi Complex. I'm not sure if that's the way a lot do it, but that's how my teacher did. So, but Golgi Apparatus, it's this thing. Kind of looks like the smooth ER in a way. Oh, sorry, the ER in a way. All right, here we go. We have already mentioned that vesicles can bud from the ER and transport their contents elsewhere. But where do the vesicles go? Before reaching their final destination, the lipids or proteins within the transport vesicles still need sorting, packaging, and tagging so that they end up in the right place. Sorting, tagging, packaging, and distributing lipids and proteins take place in the Golgi apparatus, also called the Golgi body, or as I've been called again, the Golgi complex, which is a series of flattened membranes. Alright, this is probably obvious to everyone, but when it says like packaging or distributing, it doesn't literally mean put into a box and send it on a truck. That's obviously just describing it in an analogy. But it is important to understand some of what it means. For instance, when it says packaging, it means like putting into one of the vesicles, or tagging, it means like putting something like a spike protein on it. When we go into things like blood cells and viruses, you'll figure out that things like tagging proteins, or identification proteins actually, are extremely important to life, and I don't think life could function without them. So I just want to let you know, so I just wanted to go over why that's important. Okay, here we go. We call the Golgi apparatus the cis face, and the opposite side is the trans face. I believe cis means to stay the same and trans means to change. I'm trying to see if that's correct. On. Yeah, I think so. Or yeah, I think it means like, or meaning like with or against something that I can't remember. It's not quite how we like. Obviously, when you hear things like cis and trans, you think of another subject. But in science, I think it means like with or against, or like stays the same or to change. I'll have to look that up. To transport vesicles that form from the ER travel to the cis face, fuse with it, and empty their contents into the Golgi Apparatus lumen. As the proteins and lipids travel through the Golgi, they undergo further modification to allow them to be sorted. The most frequent modification is adding short sugar molecule chains. These newly modified proteins and lipids then tag with phosphate groups or other small molecules in order to travel to their proper destinations, hence the packaging and stuff. Finally, the modified and tagged proteins are packaged into a secretory, I can never say it properly, vesicles that bud from the Golgi's transface. While some of the vesicles deposit their contents into other cell parts where they will be used, other secretory vesicles fuse with the plasma membrane and release their contents outside the cell. So basically, what's going on here is that the proteins are being packaged and being sent into the Golgi apparatus. It's going into all of this, and then on its trans side, it will be released outside the cell. In fact, you think of Golgi apparatus as kind of a go-between. Like it's basically preparing it so it can go outside. As if it just went from the rough ER to the outside, it wouldn't be functional. It has to be functional outside the cell. Okay, lysosomes. In addition to their roles as the digestive components and organelle recycling facility of animal cells, lysosomes are part of the endomembrane system. Lysosomes also use their hydrolytic enzymes to destroy pathogens or disease-causing organisms that might enter the cell. A good example of this occurs in macrophages, a group of white blood cells which are part of your body's immune system, and a process that scientists call phagocytosis or endocytosis. A section of the macrophage's plasma membrane invaginates or folds in and engulfs the pathogen. The invaginated section with the pathogen inside then pinches itself off from the plasma membrane and becomes a vesicle. The vesicle fuses with the lysosome, and the lysosome's hydraulic enzyme then destroys the pathogen. That gives you a good insight in how basically our immune system works. And in essence, the lysosome works as the cell's immune system. In fact, like I said, a lot of the stuff you are seeing here is analogous to humans or animals in general on how their system work. Like here, the rough ER, I guess, could be like, I'm trying to think. That would be like your system that helps you grow or something, like metabolism of sorts. While the Golgi apparatus could be like your repair system or something. Obviously, it's not perfect, but you get the general idea. And that's what makes this fascinating. When you see like tiny cells, you wouldn't think that there'd be that much to talk about. But very often, the cells, while just right around this, the cells are the simplest part of an organism, of a of an animal basically, or a plant. Um, yeah, sorry, eukaryotic. And even if it's the most simple, it's still quite complex. In fact, it's so complex that it's basically its own organism, and it's the simplest. It's the simplest form of an organism. That's just wild, isn't it? Well, that's essentially all I wanted to talk about. The next subject would be on the cytoskeleton, which is not quite like our skeleton, but it is similar. In fact, I think cytoskeleton is a little more similar to our skin, but it's basically what holds the cell together. And we're going to talk about that next time. You wouldn't think there'd be a whole lot to talk about, but trust me, there is. So just want to thank you all very much for watching. Have a wonderful day, and God bless.